lab today, what's it called in your lab sheet? Size exclude, okay. Yeah. What this lab is actually about is really about protein purification. And oftentimes when you're interested in working with a certain protein, you have the challenge of how to separate a particular protein of interest away from the thousands of others. So if you remember when we were talking about molecular weight determination, where you can load total protein from a cell on a gel, and it actually makes an incredibly big column of smear of 10,000 proteins. This technique that we're doing today would let you get some proteins out of this mix away from all the others to start narrowing it down to looking at what protein are you really interested in. And there are four major ways to purify a protein that you're interested in, and 3 of them are in the background material on your handout. One way is something that I've already mentioned, is that you can engineer the protein to contain a covalent tag. For example, if this is my protein, I can engineer it to where it has a particular tag called 6X his, which is just 6 histidines literally in a row on the end of the protein. And then I can use antibody techniques to bind to this and then pull this protein out from everything else. So I engineer it artificially to have this tag, and then I use this tag as a handle to bind this section onto some kind of antibody and like fish out that protein like fishing a fish out of a pond. This is the bait, so to speak. That's one way. The other ways, which are listed on your handout again, are various types of column chromatography or column purifications. And one good thing about this kit is I think that the material, the background material that they provide is really good and understandable. So there are 3 main types of column purifications, and in all of them you are making some kind of column, usually in a syringe type apparatus, to where you are loading total protein into the top of this column, and something in the column matrix, or you can think of as what material is in this column, something in the column matrix is preferentially trapping what you're interested in. So you can load stuff into a column, a lot of the liquid will flow through, hopefully the column is trapping what you're most interested in, and then you can use some kind of trick after all of the uninteresting parts have flowed through, and maybe you've thrown them away, or maybe you just set them aside. You can use some kind of trick to release what you originally trapped in the column. And there are 3 different styles of doing this. The first style, actually, maybe I'm not going to write too much about these since you can unlist it in your notes, but to just draw what they are. So the first style is gel filtration or size exclusion, and that's the type that we're actually doing today. And in this one, the stuff that is in the column matrix are beads with particular sized channels running through them. So you've got particular beads, they're often made out of glass. That's what is making the column matrix. And they have channels of a certain diameter that are running through the bead. And when you have a bunch of these together in a column, stuff that is smaller than the channel diameter will go on in there. So if the diameter is D, anything that is smaller than D will go through the channels and get stuck. Anything that is bigger than D, I need a different color here. Let's make this a different color. Anything that's bigger than D will go between and flow through fast. Stuff that is small enough to actually enter these channels or these tunnels in the beads. These will go through slowly. It may go through there. It could end up entering this one next. And it comes out slowly. All of these column purifications are somewhat like a race. So everything goes in at the top at the same time together. Some stuff is coming out fast. In this case, the larger items that can't fit in those holes come out fast. The little stuff that fits through the tunnels gets hung up in the tunnels and comes out more slowly. So when you're doing these columns, how many of you did these in biochem? Who was in biochem? Okay, good. Does every team have a biochem person on it? No? It's going to be okay. I'll come back there and stand with you. The only weird thing about this is the flow through. So when you're collecting the flow through, you're going to put all the stuff in. It starts flowing through, you do some washes, and it keeps dripping out this end unless you put the lid on. There are certain times when you put the lid on, certain times when you take it off. And the flow throughs, you catch them in some little tubes or some little beakers or whatever. And when you catch these, they're called fractions. Like a fraction of what you put in at the top. Somebody's going to be holding the tube under there, or you can prop the column on top of the tube, drip, 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 and like 5 drips, that's fraction 1. The next 5 drips, that's fraction 2. So usually the fractions from time 0, I think you've got 10 today or something like that, up through time 10. What you catch here is the slower stuff. What you catch here is the faster stuff. So a lot of columns, you put the protein in, let it settle through, and then you put some wash buffer on the top to wash it on down, and then you're catching little catches or little fractions of what drips through in time order. So in this scenario, where are the large proteins going to come out? In which kinds of fractions? The large ones are going to come out fast because they go around the beads because they can't fit through the holes. Larger stuff's going to come out fast, smaller stuff's going to come out slower. So that's situation 1, that's the one we're using today. I want to talk about the others as well because you might end up using those in your future. So method 2 is still using a column. We're still collecting fractions. The fractions are still sort of arranged in what goes through quickly to what goes through slowly. But in this situation, it's called affinity chromatography. And affinity, you probably know, is one of my favorite words. This binding affinity, and this tight binding, and this less tight binding, and a high binding affinity, and a low binding affinity. And by affinity, you can think of non-covalent binding, which depends on those electrostatic surfaces and size matching and so on. In this method, you are catching protein of interest putting its binding partner, or let's say linking its binding partner to a bead. So you still have a column, and what's in the column matrix is still beads, but this time the beads are on a covalent linkage to whatever the binding partner is. So one that I've used in the past in this class was having glucose linked onto the bead. What can you catch with this? If this is glucose linked onto a bead, what can you catch with it? What would have affinity for that? Maybe. I wasn't going for a real specific answer about like that. Actually, I don't know if insulin binds directly to glucose. But if it did, you could catch it in this method. In this method, you can catch any protein that binds glucose. And there's more than one of those. And this is what the method I've done in the past in this class. It didn't work too well, so I'm switching to a new one this year. But in this case, you put your proteins in, you've got a matrix of beads with a bait on it. Anything that binds glucose will get trapped in the column. This is the cool part. And this is an important word I didn't say yet. You elute the column, or get the protein back out. This means untrap, release, get the protein back off the column. How could you get it off the column?
Wash with a glucose solution.
Maybe even this. You just put a lot more glucose in the column, and there's more glucose floating in the column than there is glucose stuck to the beads. And the glucose replaces or outcompetes this. You just add, actually didn't put, I don't know what this is, it's been in here for a while. It may not be glucose. To get things back off the column, you would add excess binding partner. To release the protein from the beads. And then later on you may get fractions of, when your protein that you're interested in comes off, it's going to be mixed with glucose, but you can get rid of the glucose later. Because by trapping it in the first place, you got rid of every protein that doesn't bind glucose. And then you wash those away, and then you add glucose in the top of the column. It outcompetes, replaces this binding event, and your protein of interest can come on through. That's affinity chromatography. One other method that I have not tried myself is called ion exchange. And this one to me acts a lot like affinity chromatography. In this one, the bait or the beads are using charged particles to trap the protein of interest. Like, for example, here's the bead. It's putting some big charged thing out here like phosphate. I don't know if that's a real example, but hang some phosphates out here that are negatively charged. What will this trap? What will this catch? Positively charged stuff. That. Okay, it caught positively charged stuff. Negatively charged stuff will keep going through. To get this off, after you trap it, you would elute with anions. This one to me doesn't seem as useful because stuff can be positively charged or stuff can be negatively charged. This may not be a useful first pass solution where you dump all the proteins in the cell into the column and you're going to get out half of them. This might be a useful step for further purification. But it works the same way. And generally, any of these methods that you're using, generally, if you're doing this right, the first fractions don't so much contain the protein of interest because you were trying to trap it on the way down in the first place. So usually the very first fractions, time 0, 1, 2, 3, those aren't usually the precious ones that have what you want, usually. It's usually the later fractions that have what you want. So our experiment today, if you will turn to page, maybe the third page, in the chromatography lab. Carrie, my work study student, has set out and has already made a bunch of materials for you, so this is going to go fairly fast once you get started. So what you're getting is each three, you're going back to your groups of three. Each three is getting a column, and it kind of looks like a syringe, and it has a twist-off thing right here, and it has a cap. And so as soon as you twist this end off, this is going to drip, and the instructions tell you how long you let it drip and when you put the top cap back on and so on. So you're getting a column, that's in the checklist. It's called sizing chromatography column. You're supposed to have one of those. You've got 12 little tubes for fractions, and Carrie has portioned those out for you. When you twist off this thing, that opens the column, and then you've got a yellow cap that when you don't want this to drip into a tube, you put that yellow cap on. That's like a stopper. You're getting one of those in your group. You're going to use micropipetters to do your transfers. You already have a marker, and your collection tubes are in the tube rack. And the other things that you need, you're getting a protein mix. And in your protein mix, it has two different things. It has hemoglobin again, and it has vitamin B12. And these each have individual colors. In the tube, I think that they're a reddish-brown color together, and each one of these has its individual color that you'll be able to see in the fraction tubes as they come out. Which one of these is big and which is small? This is not even a protein. This is a little vitamin thing. This is the small thing. This is the big thing. In your protocol, it's describing what their colors are. Hemoglobin is always usually red. I don't remember what color this is, but it describes it for you in there. So you're going to be following the directions laboratory protocol, and these are quite easy, and the only major change is you're not going to use plastic squeezy pipettes. You're going to use micropipetters to do this work. And you're going to be collecting fractions, and then in the end you're going to have 10 collection tubes, and the other 2 tubes you're using for, actually you really only need 11 tubes. One's for waste and one's for buffer, and you're getting a falcon tube with buffer in it. You're going to have 10 fractions, and hopefully you will have separated hemoglobin from vitamin B12. You've also got questions at the back of this. So because this is a really easy lab and because there's not that much to do, I want you to turn in these questions with your lab book when you turn it in. And the back page, it's only the bottom 3 questions with the asterisks.
You want those in our line real quick?
Just turn these in.
Probably do the questions on, probably on this paper. Or I guess you could just write them into your lab notebook. Maybe that would be better. It doesn't matter to me, as long as I get them back. But the questions will become part of the points for the lab book.
So if we attach it, they just want us to put it in like results and be like, attached.
These would probably come after the conclusions,
Okay.
actually. What's your data gonna be this time? Picture of the tubes. The colors are gonna matter this time. So either get a color printout and put in your lab book, or get some color pencils and just draw a picture nicely of what the fractions generally looks like. Either way will work. And I haven't seen this particular lab before, so we'll see. Should be good. Other questions? Yeah, turn that off.